Good day to our participants. We welcome you again to MCC International Webinar Series. This is brought to you by the Mabalacat City College, Office of the Vice President for External Affairs, and the Institute of Arts, Sciences, and Teacher Education. Our topic for today is on metabolic engineering of Corynebacterium glutamicum for the synthesis of platform chemicals and polymers. Our speaker for today is Ms. Kayan Baritugo. She is a PhD candidate at the Division of Chemical Engineering and Material Science at Ewa Women's University, South Korea.
So in this talk, I will present four key topics. Firstly, biorefinery, its definition, white biotechnology, and some synthetic biology and metabolic engineering. The second topic is nylon, the definition and applications, related monomers, polyamines, amino acids, and biochemical synthesis. And then we will concentrate on the strain that I use, Corynebacterium glutamicum, and how it is ideal for bio-based nylon production, and then the specific product that we'll focus on is Cadaverine and its applications and biosynthesis. A biorefinery is a facility that integrates biomass conversion process to produce fuels, power, and value-added chemicals from biomass. A biorefinery is similar to an oil or petroleum refinery in terms of the production of multiple fuel chemical products and materials. However, a biorefinery is better because it uses various components of biomass and their intermediates, therefore maximizing the value of the biomass-derived feedstock, and also it is a carbon neutral process. So under the biorefinery concept, biomass is transformed into fuels, chemicals, and materials, and also as a combined heat and power source by two platforms. The first platform is the biochemical platform or sugar platform, wherein biorefineries break down biomass into different types of component sugars for fermentation or other biological processing into various fuels and chemicals. On the other hand, thermochemical biorefineries transform biomass into synthesis gas or syngas, which is mostly composed of hydrogen and carbon dioxide. Now, this syngas can be used as a pyrolysis oil. In the thermochemical biomass conversion, it's more complex and uses components, configurations, and operating conditions that are more typical of a petroleum refinery. I mostly work with the when biomass is converted into syngas. Syngas is then converted into ethanol-rich mixture, which can be used as a biofuel. It involves three main processes. The first is sugar production from biomass, which is the biomass feedstock pretreatment stage. The second process is the microbial fermentation process, wherein microorganisms utilize the sugars from biomass to produce the target chemicals and polymers. And then in the third process, it is the purification and further valorization of the target product which we have produced from the fermentation stage. Now, during the third process, or the separation, recovery, and purification stage, it involves very different types of processes as shown in this figure. Now, after this purification stage, that is when we get our building block chemicals or platform chemicals. Now, we call them that because they are like bigger, bulkier chemicals, which more products can be formed from. So why biorefinery? Why are we developing this process? Well, it's mainly to save the earth and for us to have less independence on already depleting fossil fuel reserves and to have a lesser carbon footprint because petroleum refinery processes produces a lot of harmful gases and chemicals which further exacerbates global warming. The biorefinery concept is further realized by using white biotechnology. White biotechnology or industrial biotechnology is the kind applied to industrial and other production processes. It's the application of biology and chemistry for industrial purposes, including manufacturing of fuels, chemicals, and polymers from biomass. The three other types of biotechnology I'll just show here. Red is for medical biotech, white for industrial biotech, as I've just said, blue for marine biotech, and green for agricultural biotech. This is an example of a biorefinery for ethanol production from sugarcane. The sugarcane biomass is firstly pre-treated. Now there are three main forms or types of pre-treatment. So the biomass is firstly physical pre-treatment, which is when the biomass is like harvested or milled or like cut from the crop. And then there is the chemical or biological pre-treatment process. They can be separately used, but they can also be used together. So chemical pre-treatment involves ammonia in this biorefinery, and the biological treatment is the enzymes. Now the enzymes here are the purified form from bacteria. They're usually involved, or they are enzymes for starch to sugar conversion. Or if the biomass is lignocellulose, it will be enzymes that are involved in breaking down lignin and cellulase components of lignocellulose. So cellulase type of enzymes. Now for this biorefinery, starch is mainly converted into glucose. So glucose is then funneled into the fermentation process, which uses, in this example, glucose from biomass is transformed into ethanol by yeast. Yeast, this can be Saccharomyces cerevisiae or Zymomonas mobilis. Yeah, I think these two strains are mainly involved in ethanol production. Now, the ethanol produced in this process is still mixed in with the culture medium and cell debris in the fermentation process. So, to get ethanol, it has to be distilled and dehydrated, so purified from the fermentation process. So, this is the downstream process. Now, during the downstream process, there is whole stillage production or whole stillage residue from this process. So that is separated, and it is also transformed into thin stillage, and syrup, and wet cake. Now, the wet cake, we can use it as high protein stock feed for animals. And the thin stillage and syrup are evaporated and not used anymore. So the purified ethanol, the purity is up to like 99.8%. So which is good for a biorefinery process. So now that ethanol, that high grade ethanol is now used for under the areas of white biotechnology, or the products from white biotechnology are chemicals, polymers, and fuel. Under the biochemicals, the process is cost effective and it offers processes that may not use chemicals. The already established biorefineries for chemical production are from DuPont, Purac, Novozyme, and Genomatica, and they produce 1,3-pentanediol, D-lactide, acrylic acid, and 1,4-butanediol. Now, all these chemicals are considered platform or bulk chemicals because it can be further used for production of other smaller chemicals. Now, for biopolymers, the polymers produced by DuPont, NatureWorks, Purac, and Sulzer, and Telis, they are environmentally friendly and biocompatible. So these PTT, PLA, and PHBV are polylactic acid based polymer, which is based from lactic acid, and PHBV or polyhydroxybutyrate valerate polymer, which is a type of polyhydroxyalkanoate, mainly derived from Ralstonia eutropha. This kind of polymer is also biodegradable, so it's great for the environment. For biofuels, the biofuels produced under white biotechnology are mainly ethanol, isobutanol, jet fuel, and fatty acid-based fuel. These fuels have higher energy efficiency and also allows decreased carbon dioxide emission. Main companies that produce biofuels are Petrobras, Gevo, Amyris, and LS9. This figure shows what building blocks and products can be produced from natural gas processes and crude oil or petroleum refineries. Now, the main building blocks from these two feedstocks are syngas or synthetic gas, methanol, ethylene propylene, naphtha gas oils, and these are further used for these other products like acetic acid, butanediol, C2, which stands for chemicals that have two carbon backbones to it. So we mainly refer to them as C2 or C3 type of products. Then there are benzenes, cumene, cylohexane. These are mainly used for nylon production or nylon monomers. And then we have fuels like gasoline, diesel, and jet fuel, and also asphalt. Now these are mainly what is already produced from already established petrochemical processes. The next figure shows the biomass feedstock, the related intermediate platforms, building blocks, secondary chemicals, intermediates, and products produced from those feedstocks. It is very complicated. So, main biomass feedstocks are starch, hemicellulose and cellulose, and lignin, all three which are mainly from lignocellulose biomass or plants. And we have oil and proteins. Yeah. The intermediate platforms, these are bio-based syngas and the sugars from the biomass. And then the building blocks are the platform chemicals produced from them. So for us to have a cost-efficient biorefinery, we need to have a very good microbial cell factory. What I mean by microbial cell factory is a microorganism that is capable of producing our target product and utilizing our target substrate very well. Now, in order for us to have this efficient microbial cell factory or MCF, we utilize metabolic engineering and synthetic biology principles and techniques. Now, metabolic engineering is mainly involved in the re-engineering of metabolic networks of a specific strain or cell factory. It can be used as a way to improve cellular properties such as substrate utilization, chemical production, yield and productivity, and improvement of cellular robustness or its tolerance to toxic compounds or harsh environments. Now, synthetic biology is used for the design and construction of genetic circuits and biological systems which we use to engineer the cell. So synthetic biology is mainly involved in the production of genetic circuits or a plasmid, which would allow us to apply metabolic engineering to our strain. In this figure, it shows us how we develop and improve a strain for chemical production. Now, firstly, we select a strain that may or may not produce our target product, a strain that may or may not produce our target product's main precursor, and a strain that may or may not use our target feedstock. Now I say may or may not because it doesn't matter what strain we select. As long as we have the pathway in mind, we can apply that to any host strain that we choose. So if we have already chosen a pathway that is, for example, glucose to cadaverine production. Cadaverine is a polyamine or mainly used as a monomer for nylon production. So for example, we have glucose to cadaverine in mind. We can choose to use a strain that naturally produces it, like E. coli, and we can engineer that using metabolic engineering tools. So it naturally produces cadaverine, but it doesn't produce it well because it has a low production of the precursor L-lysine. So we can engineer E. coli to overproduce L-lysine first, so that we will have an efficient production of cadaverine. Now, if further improvement of E. coli for cadaverine production is unsuccessful, we can use another host strain, for example, Corynebacterium glutamicum, which already has a good overproduction of its precursor, of cadaverine precursor L-lysine, but it doesn't produce cadaverine at all. So we have to get the key enzyme for cadaverine production, lysine decarboxylase, from E. coli, and then express it in Coraine. So we express it by using a plasmid. I'll explain the plasmid later in this paper, in this study, in this presentation. So now that we have that, we have the mutant strain. We will use it for fermentation. Now, in the fermentation part, we can do it shake flask first. Now for shake flask, it's just using a flask culture, or an Erlenmeyer flask. You can use that. So we put that in a in a shake flask machine, put it to the desired temperature and desired rotation per minute for the shaking, and then we just take samples from it. For example, for my strain, because it's an obligate aerob, so I only put 20 ml in a 250 ml flask. I only put 20 ml of the culture medium, and I put 2 ml of my pre-culture into it. So the 20 ml in the 250 ml flask allows for a big headspace for air to for oxygen to be in the culture. Now, if I use E. coli, I can do 100 ml of culture medium into a 250 ml flask, because it's not really that sensitive to oxygen. So that's why I can do that for the shake flask. Now for scale up, there is batch fermentation and a fed batch fermentation. For batch fermentation, you have a definite amount of your carbon source. For example, you have just 100 gram per liter of glucose. Now, after your fermentation, you just need to wait for the strain to eat all of that, and then you can end the fermentation. But for the fed batch fermentation, firstly you put a definite amount of your carbon source again, and then you wait for it to be at the minimum amount, like minimum of 10 or 5 gram per liter of glucose, and then you top that up with more glucose, more trace metals, more nitrogen source for the culture medium. So you top that up, and then you wait for the strain to stop eating the carbon source you keep adding to it. So it's like you ultimately wait for your strain to get full and stop eating, and then you can stop the fermentation. And then for these three culture methods, you get the sample, and then you can use, if you want to detect the target product, if it's a very small amount, you can use a mass spectrometer. And then if you have a lot, it's better to just use a high performance liquid chromatography machine. Now for an HPLC, make sure that you have the correct column in the machine for your target product, so that you're sure that the machine would detect your target product. Make sure that your column is right for it. And then After you have that data from the HPLC, you can further engineer the strain by studying the omics data or the system analysis data that you have. Now these are like general data that you would collect after repeatedly developing mutant or engineered strains. So you would have your genomics, or like all of the genes in your strain, proteomics for all the proteins, metabolomics for all the metabolites in it, and then the fluxomics would be you can have your carbon source be labeled 13C carbon, and then in the fluxomics analysis you can check how the carbons from your carbon source would be integrated into the final target product. Now, after that, if you have a very good strain already, if it's stable, and if it like always produces your product, then you can go on and like use it in a biorefinery. But if it's not, you can do this cycle again, because this cycle is the design, build, test, and learn cycle for metabolic engineering. So we go through this a lot in my lab. Now, after going through this cycle a lot, you would have your super microorganism. Now, your super microorganism would ideally be able to produce your target product without producing a lot of byproduct. It would have high yield, high productivity, and it would be very robust or very tolerant of toxic compounds and toxic environments that may occur during your fermentation. Now, for your microbial cell factory, it's the factory which transforms your substrates to your product. Now, an example that I put here in this slide is amino valerate production from glucose. Now we have that target pathway, glucose to 5-ABA. We firstly tested it in E. coli because E. coli has a lot of tools for manipulating it, and it's very well studied. So for our target product, 5-AVA, not many microorganisms would produce this, but we have studied the related literature, and we found out that Pseudomonas putida produces 5-AVA, but it produces it only in a small amount. So that's why we decided to move the key genes from Pseudomonas putida, key genes for the production of 5-AVA, from putida into E. coli. We only took the DAV-A and DAV-B genes. These genes are for lysine deoxygenase and aminovaleramidase. Now we put those genes in a plasmid. Now this is a schematic diagram of the PKE112 DAV-AB plasmid that we used to make E. coli produce 5-AVA. In the plasmid itself, we put a p-Taq promoter, which is a strong promoter for E. coli, and then in between the Taq and DAV-A, then in between gene in it, an antibiotic resistance marker, so that when we put this plasmid into a strain by heat shock or electroshock, the plasmid would stay in the cell so that the strain would produce the genes or the enzymes in the plasmid. Now, a successful transformant would be the strain with the plasmid, and it would grow on a plate with the antibiotic in it. So in an ampicillin agar plate, strains with this plasmid would grow in it, but the native strains or the strains that don't have this plasmid, they would die. So there we have it. Now in E. coli, we just put these two genes and it produced 5-aminovalerate, but it didn't produce it well. So we had to delete some competing pathways and we had to overexpress some genes so that it would produce the cadaverine precursor, L-lysine, well. So that's what we did with E. coli. Now since it didn't improve further, we tried producing cadaverine by direct fermentation. Direct fermentation is where you have your strain produce the target product from the carbon source in one culture. Now, this pathway is very good for 5-aval production using E. coli if it's used in a whole cell biocatalyst method. Now, the whole cell biocatalyst method is different from direct fermentation because in the whole cell biocatalyst method, or WCB, you will need to grow your strain first from glucose, and then after that culture, you put it in another culture where you concentrate your cells. So you just harvest your cells from the first culture, and then you put it in a reaction that would have your substrate in it. So it would involve two different setups. First is the whole cell or your strain production first, and then the cadaverine lysine to cadaverine reaction would occur in another setup. So that's the difference between WCB and direct fermentation. Now, because we want to do direct fermentation for 5-aval production, we decided to use a different host strain, Corynebacterium glutamicum, because Corynebacterium glutamicum, or Coryne, can produce a lot of lysine, the precursor for 5-aminovalerate. So we just did the same thing, but we used a different plasmid, because the PKE112 just has one replication of origin for E. coli to recognize. We used 3 different plasmids, which has 2 replication of origins for both E. coli and Coryne. So we use this plasmids which has 2 replication origins because it's easier for us to manipulate the plasmid when it's in E. coli first. Because in Coryne, transformation involves a 2-step process wherein you do heat shock first and then you do electroshock. Because Coryne is gram-negative, it's harder for the plasmid to go into the cell. Now, in this 3 plasmids, the PKCA, PEKE, and PCS208, we still put DAV-BA or DAV-AB the same way. There is a promoter, there's an RBS, and then, promoter, restriction enzyme, RBS, DAV-A, RBS, restriction enzyme, DAV-B, and then restriction enzyme, and yeah, that's it. Now I say restriction enzyme because a restriction enzyme is a specific sequence in a plasmid in which you can, you have these restriction enzymes so that when you cut the plasmid, when you put those restriction enzymes, the plasmid will be cut in this specific way, and you can have your gene fragment which would have the specific restriction enzymes on both ends, and when you do ligation, that gene would go into your plasmid. in the correct order of restriction enzyme that you have. So for example, for DAV-BA in the PCES208 plasmid, we have the BAMH1, then RBS, DAV-A, and then the NOT1. We have that gene fragment, and then we cut the plasmid with BAMH1 and N1, BAMH1 and NOT1, so that we can put the gene fragment with BAMH1 and NOT1 in it. Now when we have PCES208 with DAV-A in it, we can cut that plasmid with NOT1 so that we can add DAV-B gene fragment, which has NOT1, another RBS, and then DAV-B, and then NOT1 in it. So that gene fragment would go into the NOT1. Now, for you to check if your gene is in the plasmid correctly, we usually have it sequenced, and then when we have it sequenced, we check if it's in the correct way, if it's not upside down, if it's not, the direction is the wrong way. We can check it when we receive the DNA sequence. But yeah, that's a little bit of gene cloning for you there. So in Corine, when we put DAV-BA in it, the PCS208 plasmid was the best, and there was a good production of 5-AVA, very high compared to the use of E. coli. But there's also the byproduct of glutaric acid. So we deleted CAB-T, and glutaric acid somehow decreased, but it was still there. So this pathway still needs to be improved in Corine. Now, since a plasmid is an important synthetic biology tool for metabolic engineering, I'll discuss it further in this slide. So a plasmid is what you use. It's a synthetic biological circuit that you can use to engineer your strains. Now a plasmid would have these components in it: an origin of replication, a terminator, expression cassette, promoter, UTR, antibiotics, and antibiotics. Going back to UTR, UTR is an untranslated region. So it refers to a coding sequence on a strand of mRNA. So firstly, in this plasmid for Corine, it needs to have 2 origin of replications for both E. coli and Corine to recognize. I've mentioned it in detail earlier. Now in this figure, it would show you different replication of origins that E. coli and Corine recognizes. Now Corine can recognize these different replications of origins, and when it recognizes this specific replication of origins, or ORI, it can produce a low to high copy number of the gene. Now, it is very good to have a high copy number of your target gene if you want to produce the enzyme very well. Now, if producing the enzyme very well affects the strain negatively or affects the production process negatively, you need to use a low copy number of replication origin so that the negative effect is minimized. For promoter, it can be constitutive synthetic, inducible synthetic, or inducible foreign. Now a promoter is what would drive the translation of your gene of interest. Now in Corine, inducible foreign promoters just means that the promoters are from different strains or different microorganisms, and inducible means that if you put a specific chemical into it, it would make or increase the or signal the strain to produce this enzyme now. So the inducible chemical or the chemical to induce this type of reaction is IPTG or arabinose. Yeah, arabinose is a monosaccharide, and temperature, like if you turn the temperature higher, it would cause this promoter to activate this gene. So that's what inducible means. Inducible native is it just means that it's in the strain. And yeah, it's inducible by acetate or gluconate, these promoters presented here. Now for the constitutive synthetic, when we mean constitutive, it doesn't need the inducing chemical for the strain to produce it. So it continually activates the reaction, or it continually activates the production of your gene of interest or your enzyme of interest. For untranslated translation region, it has something to do with the RBS design or ribosome binding site design. So if you design your RBS in a specific way, it can either make the translation of your gene as efficient or less efficient. Now, as I said in the ORI, if you want efficient production of your gene, it's better to use a very good RBS design or an RBS design that your strain already recognizes. Now, if high production of this enzyme causes negative effects on the chemical production or on the strain, it's better to decrease the gene's transcription or translation by decreasing the efficiency of the RBS site. Now for expression cassette, the expression cassette would have your gene sequence in it. Now the gene sequence can either have a his tag added to it so that it's easy to purify the target enzyme, and it also allows for soluble expression of your enzyme, or you can have codon optimized genes. Now since if you take genes from different strains and put it into another strain, it is called heterologous expression. Now sometimes heterologous expression is not efficient if the codon optimization is not done. Codon optimization just means that you change the way the sequence is so that your strain can recognize it well. That's it. Now the codon optimization tool that I use is just a website, because some genes has a codon usage bias, which refers to differences in frequency of like how they have each amino acid in a specific way or polypeptide chain. For the terminator, that just means the end of the sequence, or that's just a stop or a period for your expression cassette. Now, yeah, I've fully described a plasmid. We can move on to we can now, like after describing how we successfully engineer a strain, we can now establish a biorefinery for platform chemical production. Now this figure is from my paper. It's a review paper entitled Recent Advances in Metabolic Engineering of Corynebacterium glutamicum as Platform Microorganism for Biorefinery. So this figure shows the native pathway and synthetic pathway. Native pathway is the natural carbon sources and natural products it can produce. Synthetic pathway is engineered pathways that we have introduced into this strain so that it can produce these chemicals. So you can see it can utilize a lot of different biomass and produce a lot of biochemicals, but we will just focus on one for this paper. In these slides, I just show you the key companies that are interested or already do some biorefinery methods. So for these specific chemicals, these specific routes of fermentation or chemical production is being done. Some are biological, some are combined biological and chemical processes. Now, biological part of the production process, I think, is mostly involved with the cell production and the microbial cell factory. Chemical processes usually come into play when it's time to purify the target product from the culture medium. So I'm gonna, because it's more than 30 minutes now and I'm sorry, so I'll just rush through these next slides. So nylon, it's a true synthetic textile fiber. So it's not naturally produced in nature. It's mainly produced by DuPont, and currently it's a petrochemical based product. So when we choose a product to produce in biorefinery, we usually choose a very useful product. So for example, nylon. Now for nylon is made up of several monomers such as caprolactam, hexamethylene diamine, adipic acid, cadaverine, and sebacic acid. Now these specific monomers would produce a specific nylon. So usually when we say nylon 6, nylon 6,5, nylon 5,6, or nylon 5,10, the number correlates or means that the monomer has this amount of backbone carbon in it. So since cadaverine has a backbone carbon of 5, that's why it's nylon 5. And then 10 because sebacic acid has 10 backbone carbon. These are applications of nylon. It's used in leather, footwear, apparel, automotive, and other applications. Now nylon monomers are in this slide, and nylon monomers are, the bio-based nylon monomers are polyamines. There are a lot of bio-based nylon monomers, such as some amino acids and some aminobutyrate, but I'll focus on polyamines in this slide. So polyamines are alkaline organic compounds, aliphatic, saturated carbon backbone, and at least has 2 primary amino groups. Now, the polyamines that we mainly focus on is 1,5-diaminopentane, which is cadaverine, 1,4-diaminobutane, which is putrescine, 5-aminopentanoic acid, which is 5-aminovalerate, and 4-aminobutanoic acid, which is gamma-aminobutyrate. Now, amino acids like glutamate and lysine are used for the production of C4 and C5 polyamines, like cadaverine, gamma-aminobutyric acid, and 5-aminovalerate. So, if you want to do a biorefinery for nylon production, it's best to use a strain that naturally produces glutamate and lysine as amino acids. Now, bio-based production of nylon is possible by using polyamines like cadaverine and putrescine from biomass-derived amino acids. So this is just a slide to show you the different nylon that each diamine can produce. So Cadaverine mostly involved with nylon 5s, 510, 512, and 54. It's mostly used with sebacic acid. Cadaverine polymerized with sebacic acid. Nylon 46 is from putrescine. And I think it is also polymerized with sebacic acid to produce this nylon. So, Cyanobacterium glutamicum as a biocatalyst for nylon biorefinery. So I champion this strain for nylon production because it's capable of overproducing lysine and glutamate, which are main precursors for most amino carboxylate and polyamines related to nylon production. It's good because it's well studied. There's a lot of available genomic information, a lot of genetic tools I've already described earlier, and it also has specific Cas9 techniques for manipulating it. I won't go into it further. You can just check my paper. Yeah. So because Corine can utilize a lot of substrates, it can utilize a lot of sugars, so that's why it's good for biorefinery. It's been engineered, so it's and there's a little evidence of carbon catabolite repression in Corine, so it's capable of efficient carbon or substrate utilization. In this figure, it just shows you validated CBP or consolidated bioprocess. When I say consolidated bioprocess, it just means that during the fermentation, the pretreatment part is not added to it anymore, the biomass pretreatment part, because the strain itself is capable of doing the biomass to sugars process. What I mean by that is like starch, silane, and cellulose biomass. They usually need to be pre-treated first before it's put into the fermentation process, but in a consolidated bioprocess, the microbial cell factory that we have is already capable of hydrolyzing these starch, cellulose, and silane so that it can produce the the sugars from it. So starch mainly has glucose, maltose, cellulose mainly has cellobiose and glucose, and silane usually has lignin, some xylose, arabinose into it. So we just put these necessary enzymes plus the enzymes for the target product production to make a CBP. This picture here, it shows CBP that can be done with corine in it. This figure is just to show you the chemicals that corine can produce already. Now, the picture in the middle with the black arrows, that's the natural pathway. Then the highlighted parts are they are the main intermediates from which these chemicals are from. So in this figure, it's dihydroxyacetone, which is the main precursor for 3-hydroxypropionic acid, 3HP, and 1,3-propanediol. So this is the pathway for it. Now, the enzymes here are mainly overexpressed or heterologous expression. It's the same with 2,3-butanediol production and isobutanol production. It's mainly heterologous expression or overexpression of genes. Now, these 2,3-butane-diol and isobutanol is mainly from the main intermediate from glucose utilization in the strain, which is pyruvate. Now, another advantage to using corine for biorefinery is that it's capable of the growth-arrested bioprocess chemical production. What I mean by this is that the strain is capable of continuous sugar metabolism under oxygen deprivation conditions, which leads to higher organic acid production and resistance to fermentation inhibitors. So this just means that the biomass, the cell growth phase, is separated from the production phase. Now, this is different from the whole cell biocatalyst method that I said earlier, because in this method we induce oxygen deprivation in the same fermenter. It unlike in whole cell biocatalyst where you have two separate setups. In this growth arrested bioprocess, it's still one setup. It's just you lower the oxygen supply into the fermenter so that it becomes anaerobic. Yes. So this is an example of succinic acid production under aerobic and anaerobic pathway. So yeah, this figure just shows you the active pathway under the different conditions. Now this figure is a summary of polyamine production in engineered Corynebacterium glutamicum. Because Corynea cannot naturally produce any polyamine, the heterologous expression of enzymes from E. coli and Pseudomonas butida is almost always the case in order for Corynea to produce Cadaverine, aminovalerate, glutarate, gamma aminobutyrate, and putrescine. Now, because my strain can produce a lot of polyamines, I just decided to focus on cadaverine production using my strain. So cadaverine production using my strain involves the polymerization of fermentation-derived cadaverine with adipic acid, sebacic acid, or dodecane acid. So these different chemicals, when polymerized with cadaverine, would produce different forms of polyamide 5,6, 5,10, and 5,12, which are used for these examples of applications. Now, cadaverine production strategies firstly involves just one key enzyme for the transformation of lysine to cadaverine, which is lysine decarboxylase. So in E. coli, lysine is converted to cadaverine naturally because E. coli has lysine decarboxylase gene. It actually has two lysine decarboxylase gene, the LDC and the CAD-A. The LDC is not inducible, so it's a constitutive enzyme so that E. coli can always produce cadaverine. And then CAD-A is inducible. So I think when either lysine is in the medium, that's when CAD-A is active, I think. For Corine, it needs heterologous expression of lysine decarboxylase from E. coli or other strains. Now, in this paper, we produced cadaverine from glucose using recombinant Corynebacterium glutamicum. So firstly, we did plasmid-based expression using the PCS208 plasmid, and then we wanted to find out what synthetic promoter is best for high cadaverine production. So we tested different low, intermediate, and high strength promoters. And then after we selected the best promoter to use, we integrated the best promoter and enzyme combination into the genome of Coraine by inserting the specific gene cassette into the lyse site or lysine export system. So this enables us to have genome-based expression of LDC while also deleting lysine export. So this allows us to have a strain that has enhanced lysine accumulation in the cell, which means for better cadaverine production. And after that, we purified cadaverine. We optimized it by comparing what's the best solvent for cadaverine purification. So we compared N-butanol and chloroform. And then we polymerized the fermentation-derived cadaverine with basic acid. So this is how we found out that fermentation-derived polyamine is actually good and comparable to petroleum-based cadaverine for nylon production. Now, we firstly screened the best lysine producing strain that we have to choose the best host strain. So we chose PKC because it produced a lot of lysine. And then we compared plasmid-based and genomic-based expression of LDC. And we, for flask culture, the H30 promoter was better at producing cadaverine, but for genome-based expression, it was H36 promoter. But like for flask culture, the environment for cadaverine production was not very well controlled. So we used the genome-based strains for further batch fermentation. And that's how we found out that the H30 LDC strain is the best strain for the best promoter and enzyme combination for cadaverine production. It produced 23.8 gram per liter. Now, after batch fermentation, we also, because the previous batch fermentation, we did fed batch after that. So firstly, we've tried to find out how much lysine does the strain produce so that we can compare it to the product. So since PKC can produce 140 gram per liter of lysine, it was able to produce 104 gram per liter of cadaverine when we use the strain with genomic insertion of E. coli LDC at lyse site. Now, for purification, we found out that it's better to use chloroform because we got higher extraction yield. And these are the extraction apparatus, separation vessel, and evaporation system that we used. Now, this is just a comparison of yield that we got for the upstream process and the downstream process. This is just to show you that there's a very good yield of cadaverine using the process that we did. And then, so that we were sure that it was really cadaverine that we purified, so we compared it to a concentrated and a distilled sample. And the peaks were similar, so that's why we were sure that it was cadaverine that we purified. We also did NMR analysis for it. just to make sure again. Well, I think this figure shows us because we found an unknown chemical or an unknown product in the first graph. That's why we also did further NMR to identify it. And we think it's this byproduct, but this is only during the purification process. This chemical was not present during the fermentation process. The 3-amino 2-cyclohexane. Now, so we compared material properties of the chemical-based polyamide and the bio-based polyamide. So some polymer properties were similar, some were better, but in conclusion, bio-based polyamide is it's good to use since it's a carbon neutral product. So it is possible to have sustainable nylon biorefinery. Now, in another paper, in a follow-up paper actually, we decided to test different LDCs so we would improve the yield of cadaverine from glucose. So we found out that the use of the H30 promoter is still the best promoter for cadaverine production. However, lysine decarboxylase from Hafnia alveoli was able to produce better or higher yield compared to our previous paper. And we also inserted the H30 Hafnia alveoli expression cassette into the genome in the same site, the lysine export site. And we also synthesized and compared the quality of the bio-based polyamide again. So these were the results. So the H30 promoter and E. coli lysine decarboxylase and Hafnia alveoli decarboxylase were better than all the lysine decarboxylase that we tested from these other strains. I think LS is for Lactobacillus cereumeri, Vibrio for VV. Yeah, those were the other LDC from other strains. Now this is the comparison of the previous LDC strains and the new Hafnia alveoli strains. This is the plasmid-based expression. So we did batch fermentation, and the best strain was the H30 HALDC strain with 30.8 gram per liter. The other one is 28.4 gram per liter by the H36 HALDC strain, and then followed by PH30 and PH36, which are the E. coli, the Crinus strains with E. coli LDC in it. Now we inserted, for this one, we still compared plasmid-based expression of the previous strain and the new strain with E. coli LDC and Hafnia LDC in plasmid. So the best titer was 93.7 by the plasmid-based expression of Hafnia alveoli LDC under the H30 promoter. Now we inserted the Hafnia alveoli LDC into the lyse-E lysine export site in Corine, and the highest titer that we got was after fed batch fermentation, which produced 125 gram per liter of cadaverine, 21% higher than the previous 104 gram per liter, with the same kind of strain, but just different enzyme, E. coli LDC enzyme. And this is the properties of the bio-based polyamide, and we just compared it again to the chemical-based polyamide. Most of the properties were similar, so just means that it's good to use. So it's good to further develop a biorefinery based on bio-based cadaverine and polyamide production. Thanks for listening. This is my ResearchGate site where you can have some of my papers. You can message me there too if you have questions. This is my lab website, which has my professor and my lab mates in it from Iowa University. Anyway, thank you. This has been a very, very long talk. I see it's like 52 minutes long, so I'm sorry. But also, thank you for listening. Bye.
On behalf of Mabalacat City College OIC President, Honorable Mayor Crisostomo C. Garbo, and the Chief Administrative Officer of MCC, Ms. Michelle Aguilar Ong, we would like to thank Ms. Kayan Baritugo for sharing her time and expertise as our resource speaker for today. For the last part of our webinar episode for today, we invite everyone to please answer the online evaluation and feedback form within today, July 25, to receive your e-certificate. The form will be closed at 12 midnight of July 26. Thank you, and we hope to see you again soon.